today we're talking about how to do a nested PCR in our lab. So the first thing you need to do is to go online and create a PCR template or follow your PCR template. And after you filled it out, make sure to print it out, put in your lab notebook like here. So in this orange area, you are going to put the number of samples you plan to run plus 2 negatives up here. So if I'm running 10 samples, I'm adding 2 more up here for our negatives. This autofills down here for our PCR error or pipette error. And everything else should autofill as well. I still like to double check these equations, or at least eyeball them over, because sometimes the equation gets messed up on your Excel spreadsheet, which will mess up your master mix, which messes up your PCR. And you won't find that out till the end, so just make sure that this is all correct. Also make sure you're using the right template for the right stage that you're on. You can see up here, this is your initial PCR template, so don't use your initial PCR template on a nested because the quantities are different. Something else you need to keep in mind while you are making your master mix is you want to add the reagents in order that they appear on the template. So your H2O, 10x buffer, DNTPs, forward and reverse primers, and your Takara tack. This is particularly in this order because it goes from the cheapest reagent to the most expensive. So if you mess up, which totally happens sometimes, it's fine, but hopefully you mess up after the H2O or after the 10x buffer, because those are cheaper reagents and you don't want to throw out our tack or the most expensive reagents. The other type of PCR we do in the lab is called a qPCR, so there are some key differences. qPCR measures fluorescence, and you have a computer that it shows you a curve. Our nested PCR, we don't have a probe, we don't have the fluorescence to measure. We're trying to obtain a sequence. So after our nested PCR, the final step of the PCR, you run a gel. If it's positive, then hopefully you're going to get a band, and that is your product that you have to clean up and send out for sequencing. So you finally obtain a sequence that will tell you exactly what you're looking at, hopefully. Another thing with the nested PCR, again, is there's 2 steps. So there's the initial PCR you have to run, and then there is the nested. So the initial PCR, the iPCR, let's say it cuts out this sequence here. I just made up these nucleotides, so don't quote me. And then your nested PCR chops out the middle, or the portion, the internal portion of that, which is why it's called a nested, because your nested PCR is nested within your iPCR. Something else to keep in mind is there are different primers for your iPCR and your NPCR. So don't use nested primers on your initial PCR. It will not work. These primers also have to be diluted down to 10 micromolars. For sending out for sequencing to ELIM, that's different. It's 3 micromolars. So everything should be clearly written on the tube. It should have, if it's the iPCR, the I primer, the initial primer, or the nested primer, and the correct concentration should be written down too. So 10 micromolars or 3 micromolars. Double check this. If these are missing from the tube, I would throw it out and make new ones. So make sure everyone's on the same page on how labeling should occur. In order to make a new primer, to dilute something down, you use the C1V1 equals C2V2 concentration or equation. So here on this side for C1V1, I wrote my stock, which is 100 micromolars. That should be written on the stock too. This is diluted down from the dried primer when you initially get it from IDT. X, because we're not really sure how many microliters of the stock we need for our desired concentration. Our desired concentration is 10 micromolars. I like to make my volume be 100 microliters, just because I need more than this, and you're going through too many frost cycles on your primers. Solve for X, and you get 10. So you want 10 microliters of your stock and 90 microliters of H2O. This is just 10 minus 100. So perfect. That will give you the desired primers, and that's everything you need in order to start your PCR. So let's get our first box. There are 2 different boxes for your initial and your nested PCR. Both of these have separate reagents, so if something gets contaminated, it's easier for you to keep track. The correct primers should be in each box too, so you should have your initial primers in your initial box. I still like to check what's written on the tube, because sometimes people get confused and things get moved around, so just always double check things. Let's keep our initial box out for now. Okay, so in our initial box, we have everything we need. We have our H2O, our 10x buffer, DNTPs, and our forward and reverse primers. So here's our 5S primer, our reverse. There's no N next to it, so we know it's our initial reverse primer, and 10 micromolars. I also like to write that on the side as well, just in case things get messed up. We also have our tack in here, but I like to leave our tack in the box till the last moment because we don't want to risk contaminating the tack. The entire tack and the Takara tack's super expensive, so just leave that in the box till the last moment. The next thing you want to do is to get some strip tubes for the correct number of samples you plan on running. Put them into the frozen ice pack, and I like to pop up open all of the tops. So after you make your master mix, you follow your list, you are going to put the correct amount of master mix into each of these strip tubes. So for the initial PCR, definitely check your template. For your initial PCR, at least for the 5S, you're going to add 22 microliters of master mix and 3 microliters of DNA. So let's say we went through and added our 22 microliters of master mix into each of these. Then we are going to make sure to close our strip tubes from the top. Never touch the inside. You don't want to risk contamination, just in case something's on your gloves. And go onto the top of your tubes after your master mix is in and write down which sample is going to go where. So I like to write IPCR in the corner with the date, and here are our 2 negatives, our NI, NI, because that's the negative initial, negative initial. Your numbers should correspond to the template that you made earlier. So those are your sample numbers and your tube numbers. Make sure they correspond. After your master mix is in, you are going to grab your samples. Let's say this is sample number 1. It's going to go into tube number 1. You're going to open up tube number 1 with your nail on the corner. Make sure you don't touch the top again. You'll go inside your tube number 1, pipette up and down 3 microliters, and put 3 microliters into your strip tube. Put sample number 1 away, close the top carefully. Go to the second one and repeat that process all the way down to your negatives. Your negatives, you're going to add 3 microliters of H2O. Once you're done, you're going to make sure your lids are all nice and securely closed. I like to double check that they're pressed down. Take my tube, spin your tube down in this strip tube mini centrifuge, and put this into your thermocycler. Your thermocycler will run for about 2 hours, and while that's going, or near like the last 10 minutes, or you can even do this days later, it doesn't matter, just make sure your IPCR is in the fridge once it's done. You're going to make another strip tube and another master mix for your nested PCR. So again, repeat the entire process. We're going to go in and make sure that we pull up the nested PCR template, so it says nested at the top. Another thing I like to make sure is DNTPs, forward and reverse primers, all of these quantities should be the same. So just eyeball that your equation worked and that everything autofilled to be the same down here. You're going to repeat all of your steps here, and you're going to put your tube and your tick numbers or whatever numbers you have. Plus you're going to add 2 more samples for 2 more negatives. So you should have 4 negatives at the end of the day. 2 initial and 2 nested. So let's say our IPCR is almost completed in the thermocycler. We made our nested master mix. Now we want to make new strip tubes. So again, pop up open all your strip tubes and put the right amount of master mix in here. So for 5S, you are now adding 24 microliters of master mix because you're going to do 1 microliter of product. We added them all in, and here are our 2 samples added on for our negative nesteds. Close all your tubes nice and securely. We're going to write down what this is. So NPCR plus the date. Just FYI, you shouldn't hold things like this. I'm just doing it for the camera. And the right numbers. So here's NI, NI, NN, NN. So there's our 4 negatives because this is our nested PCR. When we're ready, when the master mix is made, when your tubes are nice and closed, at that point you can go get your product from your initial. You have to be really careful with the initial because this is amplified DNA and there's a high chance of contamination. Make sure you're in the right hood. We want to be in the dirty hood, not our clean hoods. So here is, let's say, our iPCR. We are just going to add 1 microliter into each of the corresponding tubes. So 1 microliter, you would open up your iPCR, go in, pipette up and down, go to your NPCR and put your 1 microliter in. You do this for all of the ones on your initial. For your negative nesteds, we're just putting 1 microliter of nuclease-free water. After your negative are done, or after your nested product is put in, your samples are put in, you're putting this into the thermocycler, and you're running that for another 2 hours. Again, it doesn't have to be all done in the same day. It can be broken up. Just make sure you put your iPCR in the fridge in the dirty room once it's completed. So after that's done, yay, we're done with the PCR step. After that, you're pulling that out of the thermocycler, putting it in the fridge, clearly label it, and you're gonna be running a gel on that to see if there's product. So that's pretty much it for our PCR and how it's done. I will see you for the next video, which is showing us how to do gels.